We are recording. We are live. Man, I missed the freestyle. Now no one will ever know it happened. Except you. You were there for it. It was real. All right. So Seganet Kalamu is the profile we are doing today. And she is an Ethiopian molecular plant pathologist whose cutting edge research is dedicated to helping the world's farmers grow more food and rise out of poverty. A lot of her research has focused on elucidating different molecular determinants of host pathogen interactions, and this includes using that research to develop novel plant disease control strategies. So think about pests and the impact of pesticides on plant and crop production, as well as plant development. And so both of those areas have huge impacts on the ability for farmers to produce food. She's been awarded many different honors, including the Heroes in the Field Award by Bill Gates, who honors individuals using their talents to fight poverty, hunger, and disease. She was also recognized as one of the Forbes Africa Top 100 Most Influential Women. And there is an article that, a recent article that she has published on the desert locust and how its breeding regions can impact crop development. And I included some additional links there as well if you'd like to check out more. Have a few announcements for today. So I have decided to extend the exam resubmission deadlines to this Saturday. If you have been to my office hours, sorry, success hours, we're succeeding here, you know that they have been very busy and it's been great. It was not like this for the first couple weeks, but people are coming to talk about exams, learn, concepts, figure out tricky parts of problems, and I really am invested in your success. So the main reason that I have a 1 week deadline to resubmit your revisions is just so I can get the grades back to you, so you sort of know where you're standing before you go into the next exam. But I think it's more important that students are able to come chat with me if they want to chat, come to success hours to look over things. And, there's 71, 72 students in this course, and I don't want to rush any of the meetings. So, we were, making good progress. We're learning together, and ultimately, as long as we're learning together, I think we're succeeding together, right? So I think the smart thing to do here is to just delay that to this Saturday. So I already did one success hour session this week. The next one will be this Thursday from 10 to 11 a.m. So I encourage you to come to that if you haven't. I also have a couple more individual appointments outside of those success hours. So email me if you would like to take advantage of those. So that's sort of the key takeaway here. And then finally, one point of clarification. I don't know how this got started, but some students heard or thought maybe that only if you scored below an 80 out of 100 are you allowed to resubmit your exams. But as I was talking to some MICR 101, I've been seeing MICR 101 students in person, so that's been fun. Not like safely just happened, to drop by some of the labs in Duncan Hall. So that's been really fun, and everything's safe. But they were mentioning that they thought that only 80 out of 100 or below was allowed to resubmit, and that's not true. Anyone can resubmit. So The chat is too funny right now. If you got a 98 out of 100, you can resubmit. If you got a 99 out of 100, you can resubmit. The only score that can't resubmit is 100 out of 100, right? Like you didn't miss anything. But no matter what score you got, basically, you should be able to resubmit. And so you have until this Saturday. So those are hopefully clear policies now. Are there any questions about these announcements or the exam? Okay, great. So then just reminder to log into iClicker if you haven't already. And then thank you all for everyone who filled out the feedback. I paraphrase some of the key quotes here. The lecture portion is good. I ask good questions to the class to make sure they're paying attention. Enjoy attending lecture, opportunity to learn, things like that. So generally people are enjoying the lectures, enjoying the class. Some positive comments about the joke. I'm very touched. I don't think they're dad jokes, but we can agree to disagree on that. For the exam question we have, do we include pictures of our old answers for exam revisions? Yes, so in the Canvas portal, it says take a screenshot of, yes, uncle jokes. Take a screenshot of what you missed and then put your new answer on that right below it. And then that'll make it very straightforward for me to grade. And then another key theme was, you're welcome, Carly. Another key theme was comfortable. People feel really comfortable in this class, and that's really important to me. I want people to feel comfortable in their learning environment. So if you don't feel comfortable, it's very hard to focus on learning things, right? So that was another theme from the survey. And then I was really impressed. People appear to be liking the micro boards more than I thought, and a connection to the classroom. The other theme that came up was iClicker questions and sort of class questions. One thing that would be really helpful during lecture is more iClicker questions during class. Okay, great. Then I'm glad there are not too many iClicker questions, which can be annoying. Okay, not great. I think we could have more iClickers. Okay, I'm not a big fan of iClickers. Okay, I appreciate how iClickers. All right, I think we get the idea. So there's some divergent opinions on iClickers. I'm going to try to just sort of keep the status quo for right now, but again, I will continue to provide surveys. Usually I do it the week after each exam. And so I haven't finished reading through all of the different responses yet, but I will try to continue to improve this class based on your recommendations. So do know that I really do value your input. And if you missed a survey, there will be another one, or you can always let me know at success hours. So thank you for sharing your thoughts. This class is for you, and the only way I can make it better is if you share those things. And so I really appreciate the time you took to do that. And also just know that sometimes you may not agree with your classmates. So if I don't change something, that could be the reason why. All right. So we are now going to dig into the fascinating roller coaster ride of DNA replication, right? So that was a whirlwind, but we really made it through most of the heavy stuff. I just wanted to see where the class was. Hopefully you've had a chance to review it some more since last week on the clamp down question, which I'm again, like that joke, just so simple, so good, right? But if a bacterium ran out of clamps during replication, it is most likely that which of the following would happen? So last week we had sort of mixed results. I'm going to give everyone 30 seconds to answer here, and then we're going to jump in to the rest of the material. All right, we'll do 5 more seconds here. All righty. Okay, great. So a shift towards the right answer. That's good to see. If you're still not guessing A, please come talk to me so we can go over this conceptually here. But remember that the leading strand is the one that's sort of on autopilot. The lagging strand is the one that needs this continual cycling of clamps. And I'm happy to go over that in more detail during success hours. All right, and this is just a quick return, right? So we have the leading strand. This is the one that is on autopilot. You get one clamp, you're good to go. The lagging strand is the one that is going to have this cycling with new clamps and DNA polymerase doing that jump from one stretch to the other, like Kirby from Super Smash Brothers. I was told that several students told me that they actually understood that reference. So, way to go, Super Smash Brothers. I'm glad I am not completely irrelevant with my video game knowledge. Some final comments on the elongation part is, I should do a poll for like favorite Smash Brothers character. I'll put that on Canvas at some point. Link is the right answer, just so you know. And so, also I received several comments that eye clickers are malfunctioning or batteries were dying. If that happens, just please email me and I will be able to adjust your scores online. So for the elongation phase here, we have this model, right, where there are 2 DNA polymerase molecules. And it's important to realize that there are actually 3 polymerases. In this model, there are only 2, but in the real scientific or the actual model that has been studied by scientists, there's actually 1 DNA polymerase on the leading strand and 2 on the lagging strand. And for simplicity, this 3rd polymerase is not included in this model. However, we're going to go over it for the entire rest of the class with 3 polymerases now. Are you ready? No, I'm just kidding. We're not going to do that. But I think it's always important to recognize that while models may be convenient, often science is even more complex. And that's one distinction that I think is important to acknowledge. We are now going to move to the final sort of phase here of the DNA process or DNA replication process. So you have this elongation phase, right? Hang on. Right, so this is elongation as well as termination here. And we're going to start with discussing these RNA primers. So if you remember from last week, we had these RNA primers that help facilitate the synthesis of DNA. And while RNA primers are great, they sort of jumpstart those DNA polymerases. They are problematic in that they are RNA and not DNA. And if you're replicating DNA, ultimately that's the product you want to make, right? You don't want to make DNA with some RNA sprinkled in. So how these RNA primers are removed is that polymerase 1 or RNase H, either one can cleave the RNA at any point along the hybrid RNA DNA section. And so there is this hybrid segment here that you can see in blue and I guess dark gray or black. And RNase will just come in. It's got to eat, right? And so RNase H is going to come in. It's going to find RNA. It's going to take care of that. And so the RNase will clear the RNA primers that are interspersed throughout the replicating DNA strands. Then you have DNA polymerase 1 fill the gaps using the 3' hydroxy end of the DNA fragment. Okay, so the problem with removing the RNA primers is now you have gaps, right? And you do not want gaps in your DNA. That's going to pose all sorts of problems, structural instability, and other replication complications down the line. So the polymerase 1 enzyme will repair these regions with additional DNA bases. And then you will have DNA ligase repair the phosphodiester Nick at the junction between the repair site and the DNA strand. And that together will basically fix these RNA primer regions that, while were helpful throughout the initiation and elongation process, ultimately need to be removed to preserve the integrity of the DNA. And so this process, as I think we can appreciate, is much more straightforward than some of those earlier steps, but is nonetheless instrumental in making sure that DNA replication is going to be successful. That brings us to termination. All great complicated things must come to an end, including the end of the Marvel Infinity Wars saga. Pretty complicated, but it came to an end. Pretty cool. Sorry, I watched WandaVision last night, and that's definitely in my head. But termination of DNA replication is what should be the key focus here. And oh no, hang on. Oh, sorry, hang on. I think my headphones. Sorry, let me see if my headphones. All right. Sorry about that. My headphones just died. Well, they weren't connecting. They didn't die, but we are going headphoneless. Apologies. All right. So we are now on to terminating replication. All right. And so basically what we have here are a number of terminator sequences. All right. So this is where you have terminator sequences or TER sites that are on the bacterial chromosome. And there is a protein called TUS, which is short for terminus utilization substance, which will bind to these sequences and act as a counter helicase. And basically, TUS will stop DNAB helicase activity and thus inhibit polymerization. And if you remember, DNA helicase is the one separating these DNA strands, right? So you have a counter helicase that stops DNAB activity and thus is going to inhibit the polymerization process. Now, it's a little difficult to envision, but you have those two replication forks, right? And you're synthesizing DNA simultaneously in opposite directions. But ultimately you form these two ringed katanas, and this is what will be the result of successful completed DNA replication. And they are separated by topoisomerase 4 and the proteins CERC-C and CERC-D. And both of these recognize a specific site called diff on DNA molecules, and they're able to catalyze a series of cutting and rejoining steps. And that's illustrated in the diagram here, all right? So they don't really show the proteins, which I find annoying, but the key takeaway here is that the topoisomerase 4, as well as these two additional proteins, will pass the linked chromosomes through each other by doing this cleavage and ligation reaction. And then you will resolve these two ringed DNA molecules into separate rings, which will then be separated into the two dividing cells. And in this way, you're able to partition the genetic code for inheritance to the mother and daughter cell, respectively. And so in this way, you sort of have the oldest bacterial magic trick, right? It's just like, hey, look, like, you have those two rings, then you separate them with the activity of these proteins. The next section is plasmids, but before we move on, are there any questions about the different termination slides that we have gone over? And I will, just flip through these. All right, it doesn't look like there are any questions. Question for the class. What makes the TUS protein so dangerous? And I can ask that via chat. What makes the TUS protein so dangerous? Any ideas? No. Yeah, it's found in all the Terminator sequences / movies. Duh. Oh man. I really enjoyed having some level of gullibility, but I think my days of sneaky jokes are coming to the end. On to plasmids. Terminator sequences. You have seen the Terminator movies, have you not? Right? Come on. There we go. If you haven't, don't tell me. I will probably cry. Okay. So on to plasmids. So there are two kinds of extragenomic DNA molecules that can interact with bacterial genomes. These are horizontally transferred plasmids, as well as genomes of bacteriophages. All right, so viruses that infect bacterial cells, which we'll be talking about later today. And so the transfer of genetic material in this way is very beneficial to microbes because they can acquire different genes for antibiotic resistance, metabolism, or possibly chemotaxis, et cetera, right? So or virulence factors, there's a whole range of benefits that these plasmids or extragenomic DNA can confer to these microbial species. And they are much smaller than chromosomes and found throughout every domain of life. While they're usually circular, there are linear ones that are also characterized. And importantly, they are not self-replicating. They need host proteins for their replication. The discovery of this free DNA concept was discovered back in 1928 by Dr. Frederick Griffith. And this sort of set the stage for additional discussions and experiments that highlighted the importance of transformation, which is defined as the process of importing free DNA into bacterial cells. And this transformation really laid a framework out for how microorganisms could exchange genes, right? And this is really important, again, for those reasons I mentioned. If you are in a hostile environment facing multiple stresses, the organism that has the best array of genes to combat those stresses is going to be the one that survives. And as your environment changes, you may continue to acquire more genes and pass those on to your progeny, so that from at least a colony perspective, your genes will persist in the new environment. So transformation, more broadly speaking, is something that many different cells can do. And there are a couple different flavors of transformation, all right? So some cells, some species are capable of natural transformation, and this is dependent on complexes called transformosomes. Whereas other cells need a little extra help. They need a little bit of manipulation. This is often either chemically or electrically induced. And this is basically just perturbing their membrane in a way where they will be amenable to the uptake of the DNA. So whether or not you are naturally ready to incorporate the DNA or you need a little assistance, the key concept here is that you're taking up extraneous DNA molecules that are in the surrounding environment. And there are a number of benefits to this process. DNA can be, in a sense, food, microbial food. I know you're like, I would much rather prefer a burrito or a pizza, but that is the, in certain contexts, the microbial equivalent. Also, if we think about, again, all these stresses that microbes are facing, sometimes your DNA gets damaged, or maybe there's just an inefficient DNA replication process. And so if there's damage that has not been repaired, then DNA is the perfect way to patch that up. And then finally, again, you can acquire those genes that will help you overcome the various stresses that you're facing in a particular environment. So the natural transformation in specific gram-positive organisms is usually growth phase dependent. And so you can figure out how the transformation process impacts them by looking for the development of a transformosome complex across the cell membrane. And the basic idea of this complex is that it is binding to extracellular DNA that is floating around nearby, and then it will remove one strand of the DNA and bring in the other strand through a transmembrane pore. And once the transformosome is assembled, right, then the cell is referred to as competent, right? This isn't talking about whether the cell is smart or not, right? It's whether it is ready to take up DNA. Although honestly, if you're a smart cell, you'd be ready to take up DNA because it's probably going to help you. And now that's not always the case because there's also viral DNA, but there are other mechanisms to deal with that. The key takeaway here, though, is that if you are competent, that means you're ready to import free DNA fragments and then integrate them into your genome by recombination. We're going to go over the transformation process for the best bacteria in the entire world, and that is a fact. That's not just something I'm saying. I looked it up, and Streptococcus pneumoniae is the best, the coolest, the greatest, and it has nothing to do with the fact that I study it. But I will say that while the book does a decent job sort of outlining this, I broke it down a little further with some more concrete steps. I think this helps us put the entire process together in a little more detail. So there are a number of moving factors here. We're just going to walk through them one at a time. The first is competence factor, CF. And this is an important difference between the third and fourth edition say competence factor. The fifth edition says competence stimulation peptide, or CSP. And that, I think, reflects the specific nature of the terminology for Streptococcus pneumoniae. But fundamentally, they're all basically the same thing. All right. So one cell's competence factor is another cell's competence stimulation peptide. All right. But if you look at the figures in your book, you'll see differences in the abbreviation. But don't get confused. They're the same thing. So the Streptococcus pneumoniae bacteria, as well as many other species that are naturally competent, will become competent through a quorum sensing mechanism. And that is illustrated here, where you have a single cell that is making this competence factor, specifically a precursor molecule. And if you look, you have the COMC and competence factor precursor molecule that is cleaved in an ATP dependent process. All right. So this cleavage event occurs, releasing competence factor from COMC, which is then secreted via the COM-A and COM-B proteins out into the extracellular space. So you make the precursor, cleavage happens, competence factor is secreted out to go out into the world and explore things. And that is dependent on COM-A and COM-B. Now that competence factor is out, right, more of this happens. So you have more competence factor cleavage, export. And you can imagine, as the more and more cells do this, you're going to get more and more competence factor into the extracellular environment. And you don't even have to imagine it. That's actually what happens. All right. So you have this high external level of competence factor that will eventually reach a critical threshold. And this threshold will then mean that competence factor is sufficient to interact with COM-D. And COM-D is a sensor kinase, all right, that will detect these high levels of competence factor. And this binding interaction will stimulate a phosphorylation event from COM-D on to COM-E. So COM-E is going to now be phosphorylated because of activation by COM-D, and this phosphorylation event will lead to competence factor action, okay? Now I'm going to pause here for a second because what you can see, right, is the quorum sensing theme that is underlying this. So does anyone remember what quorum sensing is? You can just drop it in the chat or use your microphone. What is quorum sensing? Cell-to-cell communication. All right. And that is correct. But can we be more specific? What is it about quorum sensing? Would you say that two cells often engage in quorum sensing? Multiple cells must be involved. Great. Would you say that quorum sensing occurs with very few cells or many cells? All right. Amy is saying many cells. What do others think? Harpreet is also chiming in with many. Yes. So quorum sensing, right? You need to reach a quorum. And that concept, right, actually is grounded in parliamentary proceedings. But for our purposes, right, you need a critical number of cells to be secreting competence factor. So you have all these cells, they're secreting competence factor, and then that will help reach that critical threshold for the sensor kinase to detect competence factor. If you only have a few scattered cells around, then it's going to be much harder to detect this signal, right? It's going to be diluted out, washed away, etc. But maybe in a really dense microbial community, such as a biofilm, then you're going to have that signal occur. And when you have that quorum sensing, right, that's what's going to lead to this phosphorylation event of COM-E. And this is going to induce COM-X transcription. And COM-X is the gene name for SIG-H, which is the alternative sigma factor. In the 5th edition of the textbook, they also call the alternative sigma factor COM-X. So again, for our purposes, COM-X and SIG-H are interchangeable. Just note that SIG-H or COM-X for the protein, that first letter is going to be capitalized, whereas for the gene, it will be lowercase. All right. Now, this induction of COM-X is important because this leads to the alternative sigma factor, which we've talked about already. We talked about alternative sigma factors and how they have distinct transcriptional programs. And so the SIG-H/COM-X transcription factor directs the transcription of the transformosome, right? How cool is that? So basically, this protein is like, let's get it together, folks. We need to build the most amazing structure ever. Actually, it's not. Well, it's pretty cool. I have a personal favorite. It's not the transformosome, but we'll get to that in Unit 4. The alternative sigma factor here is going to upregulate all these different gene components that are going to produce proteins. And now we know how all of this works. And it's going to generate the transformosome, which assembles at the membrane, okay? And the transformosome, right, it has this ability to bind extracellular DNA. It's going to degrade one strand and import the other, and then it will be able to take advantage of the free DNA that is floating around. Now, why do you think Streptococcus pneumoniae wants this upregulated when they're is a high number of cells around. And I can type that in the chat. Why do you think strep pneumo for short? Strep pneumo wants this upregulated when there are high numbers of cells around. Anyone? Sorry. This equals transformasome. Transformasome. Slight typo, but what do we think, class? Look, I even typed it all again with the transformasome in it. That's definitely deserving of an answer, in my opinion. Is the entire class going to leave me hanging right now? Not everything is a pun. I know I came out firing a lot of puns this class, but sometimes a Tuesday needs that to exchange DNA. Okay, Damini, to exchange DNA. I like it. Do you want to expand on that, or does anyone else want to chime in on that answer? Why specific? But why not? Well, how do I want to say this? I guess this higher chance of getting beneficial DNA, right? Yes, so that's sort of what I the spirit of the question is, right? So if you have strep pneumo in a biofilm of strep pneumo, right, it's a really crowded environment, so there's going to be tons of this competence factor inducing upregulation of transformasomes, but then everyone can trade with each other, right? It's very easy to get free floating DNA if you have cells that are right next to you. But if you're in a very low density environment and your closest cell is like, I don't know, a centimeter away, I mean, that's like really far for a cell. And strep pneumo does not have flagella. So their DNA has to float that entire centimeter over and then be taken up by the transformosome. I don't think so. You want to be dense culture, and that will promote the uptake of the free DNA that is floating around, right? Cells die. I'm sorry, cells die. It happens. But when they die, their DNA is going to be released. And while will be degraded to some degree, there will be fragments that other cells can pick up. The precursors are not always made. They are subject to other regulatory systems that aren't shown here. But great question. We will hopefully touch on some of those regulatory systems later in the course, but basically there are upstream pathways that drive competence factor or precursor competence factor cleavage and synthesis. We have a question: doesn't steps 4, 6 through 6 relate enable step 1? Not explicitly, right? So there are other regulatory pathways that aren't shown here that that modulate the operon upregulation versus competence factor export, or else this would just be an endless positive feedback loop. And at some point, you're going to want to stop this, or the only thing you'll do is be dedicating your resources to this single pathway, right? And so it's really important for cells to have the ability to have intersectionality between the different regulatory systems. But these are great questions. And often, as I said before, science is usually the real answers are even more complex than we anticipate. All right, so do floating genes not alter? Can you extrapolate, or can you clarify that question, Kevin? Do floating genes not alter DNA fragments? So the genes here, these operons, are involved in the construction of the transformosome, as well as the processing of the extracellular DNA. The DNA that the Streptococcus pneumoniae can uptake may alter these operons. It depends on the nature of the DNA that they uptake. So that's going to be ultimately context dependent. But another good question. Other questions on this pathway or the different steps? Ooh. So, what if the extracellular DNA is not Streptococcus pneumoniae? Totally fine. Streptococcus pneumoniae, if it's DNA, strep pneumo is interested. So, it does not have to be specifically Streptococcus pneumoniae DNA. And Kevin, you're asking when we were learning about cool cells that are not Streptococcus. Well, we just came from E. coli, so we've been with Streptococcus for all of 10 minutes, but we will be moving on shortly. If cells are constantly sharing DNA, asked Domini, doesn't that mean that they are mutating all the time? Oh. Or is it not enough to completely change a cell's makeup? Right. So there's a number of factors here. If the DNA is taken up by the cell, it needs to be able to undergo recombination with the bacterial chromosome in order to actually facilitate acquisition. So that's one thing that is important to consider. The other thing is that the DNA needs to not be degraded, right? So that if the DNA is too degraded, then the cell won't really be able to use it. And then another important thing that some of the earlier questions were alluding to is that DNA is not constantly being shared. This is, this example is in a high density or exponential phase population. So you have a ton of strep pneumo together, and this is prime time to share your DNA, right? That makes a lot of sense because there is a lot of opportunity for exchange. It's sort of similar to going to a conference or a big meeting and handing out your business card, right? You hand out your business card, you're going to get a lot of business cards. That's a great environment. If you're just in your house with your family, you're not gonna like hand out your business card, right? So like, or maybe you would, but like if you hand out your business card to your family, like unless you have a huge family, it's probably going to be, a handful of people, and then you're going to get theirs and you're like, all right, well, this is good practice for real time. So that is to illustrate that cells are not constantly doing this, and thus they're not constantly acquiring new DNA. And because they're not constantly acquiring new DNA, that's going to decrease the number of genes that they're just going to possibly acquire. But it certainly can be strategic when they do it in the right context. Your question about mutation is a little more complicated because cells can undergo mutation through a number of different mechanisms, not all of which we've had the opportunity to discuss, but errors in DNA replication can occur that can create mutations. UV rays or radiation can cause mutations, as well as transposons and a number of other factors, right? So cells undergo mutation, or mutation can often, there are forces that are consistently applying mutations to cells, but the acquisition of new DNA is only one of them. Very good questions from the group. Any other questions, since we seem to have hit a fun topic? I mean, surprise, it's microbiology, right? But all right. Well, let us then shift gears to the competent thing to do. I won't point out the pun. It's pretty subtle. All right, everyone. We went to lab earlier, and we took concentrated supernatant containing competence factor from late exponential phase wild type Streptococcus pneumoniae and added it to early exponential mutant Streptococcus pneumoniae that lacked COM-E. You measure COM-X transcription over 4 hours. What are your expected results? I'm going to put a minute on the clock and go. The answer may surprise you, or it may not surprise you at all, but 30 seconds left. Get those answers in. Five seconds. Make it count. All righty. Wow. All right. We have a diversity of answers. I want to say everyone's a winner, but I also don't want to lie. Let's see what people are thinking. Who would like to share why they voted for their answer and why? And no matter what you voted for, someone voted with you. Any ideas there? Why did you vote for what you voted for? Domini said B because if COM-E is not present, then there shouldn't be any COM-X transcription. Interesting. And Harpreet agrees. Counter arguments. May I remind both of you that B only received 34% of the vote. Anyone who would like to champion graph C? Anyone that would like to champion graph A? Anyone that would like to champion graph D? Well, in the interest of time, congrats to all who voted for graph B. That is correct. And the logic explained by Domini is also well said. Let's briefly go back here, right, to our model. If COM-E is not present, all right, so COM-E is just absent from the cell, and I will use my very cool, super cool trick that I demonstrated last week, I think, of actually deleting COM-E. Boom, gone. Then you can have all the COM-F you want, right? You can cleave it all day and you can activate COM-D, but if you're not activating COM-E, then how are you going to get COM-X transcription? It's just not going to happen. It's not going to happen. And you are adding it, right, to a culture that is early exponential. So it's just starting out. So relatively low density. So that is consistent with an inability to generate high levels of COMEX. And honestly, no matter what stage it was at, it wouldn't really matter because it doesn't have COM-E, so it's never going to be able to generate COMEX, all right? It's trying to do the impossible because it's missing an integral link in that chain. So any questions on that explanation before we move on? Does lack mean, so yeah, lack means being absent. So if it lacks it, it does not have it. So if you lack it, then it is missing from you. Another way of saying this would be, it is a mutant that contains no COM-E, for example. And if there are ever confusions about the question, whether it's during class, whether it's during class or during the exam, please reach out to me to clarify, right? Because I want to make sure that there's a clear understanding of what the question is asking. And then let's see here. Kevin's chiming in that he chose B for the same reason, but when he was thinking about it, doesn't the late exponential wild type still have COMEX transcription? Right. So that is true, but this is asking what the COM-E mutant is doing, right? You're adding it to a culture of strep pneuma that lacks COM-E. So the wild type would have high levels of COMEX transcription. It would likely be something like A or D, and then B would be the one for COM-E. And so good practice questions, right? Because I can't unfortunately make 1,000 practice questions are, yes. So these are the results for the early exponential culture, which you were adding the supernatant from the late exponential culture from. And so what happens if you change another protein? What happens if you remove another protein? What happens if you look at something else instead of COMX transcription? What if you looked at confidence factor that was in the supername, right? All these variables are things you can play with as you prepare for the next exam. And if that is something that you'd like help practicing, again, success hours are a great place to do that. All right, everyone, it's time to move on to a comic on competence. Hans is feeling rather incompetent today, right? Because like everyone else is getting the DNA. Hey, okay. Viruses, yes. I think we've heard of them. And here we go. Our virus unit, probably highly anticipated, unless maybe you're taking virology with me, in which every day we talk about viruses, or not every day, but every class. We're going to start off with some basics on viruses, and then next class we will dig into our feature on SARS-CoV-2, and we will be reading our first primary paper of the course on the SARS-CoV-2 virus. So exciting stuff. I know. I'm a little biased, but I'm owning it. So a virus is a non-cellular particle that must infect a host cell when it reproduces. All right. So that's the sort of classic definition of a virus. And this means that it's going to need to subvert the host cell's machinery to help it produce more virus particles. If it could do it on its own, then it wouldn't even need to infect the host, and it would likely be a cell. The virus particle, or virion, these terms can be used interchangeably, consists of a single nucleic acid. So there are DNA viruses and there are RNA viruses. For example, our crowd favorite right now, SARS-CoV-2, is an RNA, positive stranded RNA virus, and contains a protective protein capsid. In more complex viruses, the protective protein may also be called a head coat. Now, different types of viruses are going to infect different specific host cells, but usually the virus species or family is limited to a particular host range, and that is due to the host receptor and virus receptor interactions, right? So it's very hard for a virus to infect something it can't bind to, right? So that initial binding and attachment step is really important. Now, bacteriophages are a specific class of viruses that infect, you guessed it, bacteria, and they form what is called a plaque of lyse cells on a lawn of bacteria. So there are different viruses that infect different cell types. You have the measles virus. This is a human virus that infects human cells. You also have viruses that are plant viruses that infect plants, and that includes the tobacco mosaic virus, for example, or TMV for short. Here you can see bacteriophages infecting a host bacterium cell, and the bacteriophages being produced inside the cell before they lyse. And then in panel C here, you can make out the capsid and virus envelope of some budding viruses. This image here are the plaques that form. So these are zones of clearance. And so the idea behind this is that an individual virus started, infected a cell and then spread to adjacent cells. And that was how this zone of clearance is formed. And by counting the number of plaques, then you're able to calculate the number of virus particles in a particular sample or solution. And this is sort of the analogous way of assessing a viral titer to a bacterial titer, or CFU, so colony forming units, are for bacteria. PFU, or plaque forming units, are for viruses. So after you have infection of a cell, the real magic begins, and you have viruses creating what are called virus factories inside the cell. So that's where they recruit a number of different molecules and proteins together to create and assemble new virions or new uh, viral progeny that will then bud off from the cell and go on to infect other host cells, right? So sort of this crazy cycle that equips a virus to then go on and repeat the process all over again. And these virus factories can be tracked and have been shown to actually move throughout the cell in different regions as they are producing more of the viral progeny. Now, you have viruses that have different strategies for how they replicate. And some of the viruses out there, and there's, thousands of viruses. So not all of them do this, but some viruses do more than simply replicate within a cell. They also integrate their genomes into that of the host. And in effect, these viruses basically become part of the host organism because once they have integrated their genome into the host DNA, then the virus DNA or the virus genome, right, is now part of the host genome. And so when the host undergoes bacterial replication and DNA replication, that will be copied, right? And so then this is now in the backbone of the genetic code of the host cell. So when a virus does this in a bacterial genome, it is called a prophage. And when it is integrated into a viral genome, it is called a provirus. And so these conceptually are the same. It's just the target cell type that is different here. And when you have a permanently integrated provirus that is transmitted into the genetic code of the host cell, that is referred to as an endogenous virus. All right, so that is basically saying that the virus genetic code is with that host forever. And what is interesting is that, you think of the standard bacterial growth curves. These do not accurately predict viral replication dynamics. And it's actually quite difficult to predict specific virus families, right? So you have the prophages, you have proviruses, and these types of viruses are notoriously difficult to predict. Does anyone know why? Or they're very difficult to model mathematically. So why is it so difficult to model prophages and proviruses? Mathematica. Any guesses? It's because they're so good at integration into the host genome. Heyo. All right. That may have been a dad joke. All right. So now we will hopefully never forget what kind of viruses integrate into the host genome. We also need to appreciate that viruses or virus particles are inert, all right? So they don't have any metabolism or conversion in terms of their ability to get energy. They are dependent on the host environment. And so that is where you have the virus or virus particle just sort of in its inert state. You also have the intercellular replication process, where within a host cell, the viral gene products direct the cell's enzyme to assemble progeny viruses as virus factories. All right, so we briefly talked about that. A shout out to Dr. Singhal, who does study bacteriophages and is working very diligently on determining phage replication curves. She may have been slightly disappointed in that answer, but for the real answer, please talk to her and her lab if you'd like to find out more, or perhaps even be part of her lab. She's always looking for talented or passionate students. The viral genome is then also integrated with the host DNA, which we've just talked about, right? So there are different forms that viruses may use to interconvert across these throughout their replication cycle. Here we have the basic dichotomy between viruses and their hosts, right? So you have the virion particle, which can infect and synthesize itself within a host, and depending on the type of virus it is, it may be cytopathic, which will then lead to the lysis of the host cell. The virus could also integrate itself into the genome of the host and then be passed down from mother to daughter cell. A stressful event could then induce its excision and result in lysis at a later point. And this gets into the dichotomy of lytic versus lysogenic phages. Okay. Is a virus alive? This is a question that has perplexed biological philosophers for many years. Some say yes, some say no. I honestly just think of Game of Thrones, what is dead may never die. I don't think of viruses as alive because they're not consistent in each of their sort of like the metrics, but you could say they become alive for a brief time when they take over a host cell. We are at 4:15, so we will pick up with viral ecology on Thursday. If you have any additional questions, please shoot me an email. I do have to run to my next class, but thank you all for your participation today. And on Thursday, we will be talking about, yes, viral ecology and SARS-CoV-2. I look forward to discussing it with you there. Take care, everyone.